Tay Sachs disease. What is it? How does it work? And why is it so deadly? Tay Sachs is a rare inherited disorder that destroys nerve cells in the brain and spinal cord. A baby born with Tay Sachs develops normally in the first 3 to 6 months of life. During the next months, or even years, the baby will progressively lose the ability to see, hear, and move. A red spot will develop in the back of the child's eyes. The child will stop smiling, crawling, turning over, and reaching out for things. By age 2, the child may have seizures and become completely disabled. Death usually occurs by the time the child is 5 years old. Tay Sachs disease is caused by mutations in the hexa gene, which is located on the 15th chromosome. The hexa gene provides instructions for making part of an enzyme called hexosaminidase A, which plays a critical role in the brain and spinal cord. This enzyme is located in the lysosomes. Within the lysosomes, hexosaminidase A helps break down GM2. GM2 is pretty complicated, but here's what it is in simple terms. The G stands for ganglioside. Gangliosides are basically a group of lipids or fatty acids in the brain matter. The M stands for monocylic, mono meaning one, and cylic meaning cylic acid, which is a type of acid found in lipids. The 2 in GM2 means it was the second monocylic ganglioside discovered. We'll get into when that was later. Mutations in the hexa gene disrupt the activity of hexosaminidase A, which prevents the enzyme from breaking down GM2. As a result, the substance accumulates to toxic levels, particularly in the neurons in the brain and spinal cord. Progressive damage caused by the buildup of GM2 leads to the destruction of these neurons, which causes the symptoms of Tay Sachs disease. Tay Sachs is inherited through recessive genes, so both parents need to carry a copy of the mutated gene for their offspring to have Tay Sachs. Even if 2 parents both have the recessive gene, there is only a 25% chance their offspring will have Tay Sachs, but there is a 50% chance that their offspring will be a carrier of the disease. Also, there is a 25% chance that their offspring will be rid of the mutation completely. Now, let's talk about how it all began. In 1881, Warren Tay, a British ophthalmologist or eye disease specialist, first described a cherry red spot in the retina of the eye in a child who also had neurological problems. He recorded this in the Volume 1 edition of the Proceedings of the British Ophthalmological Society, an organization in which Tay was a founding member. Later, in Volume 4, he reported that another member of the same family had the same retinal condition. In 1896, Bernard Sachs, an American neurologist, observed extreme swelling of the neurons in autopsy tissue from affected children. Both men were describing the same disease, but it was not until the 1930s that the material causing the cherry red spot and neuronal swelling was identified as a ganglioside. The disease was recognized as an inborn error of metabolism, meaning that for some reason the body was born without the ability to metabolize the gangliosides that are still being produced. The abundance of these fatty acids are what caused the red spot and the neuronal swelling described by Tay and Sachs. In 1960, the ganglioside was given the name GM2, and in 1969, researchers discovered that GM2 buildup is caused by lack of the enzyme hexosaminidase A. This mutation, this disease, was then given the name Tay Sachs. Now, in 2018, Tay Sachs is very rare, with under 20,000 US cases per year. Treatment can help, but the condition cannot be cured and still always ends in death.